Welcome to this video in which we shall introduce ourselves to the cholinergic system. The neurotransmitter of the parasympathetic nervous system or the cholinergic system is acetylcholine. Acetylcholine is secreted from cholinergic neurons, and the secreted acetylcholine binds to cholinergic receptors. There are two types of cholinergic receptors, namely muscarinic receptors and nicotinic receptors. Stimulation of these receptors leads to mediation of cholinergic actions. Now let's take a look at a cholinergic neuron. The synthesis of acetylcholine in the cholinergic neuron begins with the uptake of choline. This takes place with the help of a transporter, and once inside the neuron, the cholinergic neuron, acetyl-CoA combines with choline to give acetylcholine. Acetylcholine is susceptible to degeneration if left alone in the cholinergic nerve terminal. It is saved from or protected from this sort of degradation by vesicular uptake. It is taken up and stored in vesicles, and when this cholinergic neuron gets stimulated, it releases the acetylcholine. Exocytosis of acetylcholine is mediated by calcium channels, and when these calcium channels open, calcium enters the cell, and this triggers the release of acetylcholine from the cholinergic nerve terminal. Now, we said that there are two main types of cholinergic receptors, muscarinic receptors and nicotinic receptors. The muscarinic receptors include M1, M2 and M3 receptors. These are the receptors that we are going to focus upon. M1 receptors are present in the autonomic ganglia. They are also present in the central nervous system, where upon stimulation it improves learning and memory or cognition. There are many cognitive enhancers that work by stimulating M1 receptors in the brain, and these include donepezil, rivastigmine, tacrine, and galantamine, the so-called cerebroselective cholinergic drugs. These are indirectly cholinergic drugs which act by inhibiting acetylcholinesterase enzyme and increasing the amount of acetylcholine available to bind to M1 receptors in the brain. Yes, so M1 receptors are also present in the gastric glands, where upon stimulation it causes the increased release of hydrochloric acid. Certain drugs have been tried which block M1 receptors in the gastric glands, like pyrenzapine, which have been used in the past for the treatment of peptic ulcer. M1 receptors are also present in the vestibular apparatus, and upon overstimulation of these receptors, motion sickness can occur. M2 receptors are present almost exclusively in the heart, and upon stimulation it causes depression of the myocardium. There is reduced force of contraction and most notably reduced heart rate. Now regarding M3 receptors, now these are present in the longitudinal fibers of the ciliary muscle, and upon stimulation of these receptors it causes contraction of these longitudinal fibers of the ciliary muscle, resulting in opening of the iridocorneal angle and increased drainage of aqueous humor, resulting in a fall in the intraocular pressure. We will see how this happens. M3 receptors are also present in the circular fibers of the ciliary muscle. Upon stimulation, it results in an overall decrease in the radius of the eyeball, resulting in the relaxation of the lens ligaments. The lens will bulge anteroposteriorly, and there is an accommodation. There is accommodation of the lens, and we shall also see how this happens. M3 receptors are present in the vascular endothelium. Upon stimulation, it causes vasodilatation. M3 receptors are seen in exocrine glands. Upon stimulation, it reduces the secretions of these exocrine glands. They are seen in the beta cells of the pancreas. Upon stimulation, they increase the secretion of insulin. M3 receptors are also present in the sphincter pupillae, and upon stimulation, it causes meiosis. We shall see how this happens. Finally, M3 receptors are present in the smooth muscles of the bronchi, and upon stimulation it causes bronchoconstriction. Now, the nicotinic receptors consist of the NM and NN receptors, of which the NM receptors are important because they are seen at the neuromuscular junction, and they are the targets of certain drugs, particularly the peripherally acting skeletal muscle relaxants, otherwise called neuromuscular blocking agents. Drugs such as succinylcholine are considered partial agonists of NM receptors, while the non-depolarizing neuromuscular blocking agents such as D-tuberculare, pancuronium, rocuronium, vecuronium, and so on, these are considered as antagonists, competitive antagonists, or concentration-dependent antagonists of acetylcholine at the NM receptor. Right. Now, to understand more clearly about the role of M3 receptors in the eye, we need to have some basic idea about how the eye looks like. Let us now consider the cut section of an eye. We are looking at a sagittal section now. What you are seeing here is the sclera, and that little projection is called the scleral spur. This here is the cornea. And when you take a sagittal section of the eye, you will get a triangular shaped structure here. It is triangular on cut section, of course. This is the ciliary body. And the ciliary body consists of ciliary muscles. The fibers in blue are the longitudinal fibers of the ciliary muscle. The fibers which are blue and in circles are called the circular fibers of the ciliary muscle. And these red colored things are the oblique fibers of the ciliary muscle. Now this here is the superior iris, and this over here would be the inferior iris. Now, these structures contain a muscle known as the sphincter pupillae in green. And in yellow, what you're seeing now is another muscle called the dilator pupillae. And obviously it is seen in both the superior iris and the inferior iris. In gray, we see the lens, which is held in place by the suspensory ligaments. There you can see the trabecular meshwork, and a little superiorly the canal of Schlemm. Now let us look at that diagram again and try to see it as a whole, including both the superior ciliary body and the inferior ciliary body. So there is the ciliary body, the superior ciliary body. Those are the longitudinal fibers of the ciliary muscle. Those are the circular fibers of the ciliary muscle. And those are the oblique fibers of the ciliary muscle. That's the iris. That would be the cornea. And what you're looking at now is an angle. It is an angle which is formed between the iris and the cornea. It is called the iridocorneal angle. And it is of very great importance in pharmacology because this is the site through which aqueous humor exits the eye. Aqueous humor is an important determinant of intraocular pressure. If this angle closes up, then the drainage of aqueous humor is impaired and intraocular pressure increases. This is dangerous in glaucoma. On the other hand, if the angle is opened up, it will cause increased drainage of aqueous humor and thus a reduction in the intraocular pressure. So that's the importance of the iridocorneal angle. Now this arrow, of course, refers to the sphincter pupillae, and that is the scleral spur. And the longitudinal fibers of the ciliary muscle are attached to the scleral spur. Okay, now what we are looking at now are the circular fibers of the ciliary muscle of the inferior ciliary body. Let us look at these two muscles now. The circular fibers of the ciliary muscle of both superior ciliary body and inferior ciliary body. Let us focus on the circular fibers of the ciliary muscle of the superior ciliary body now. Remember, this is a cut section of the eye. If the eye was not cut, if it was intact, then you can imagine these muscles coming out of the screen towards your face and then taking a U-turn and turning downwards, and then turning away from your face and again towards the screen, towards the inferior ciliary body, and then meeting the screen at the inferior ciliary body, and then piercing the inferior ciliary body at the screen and moving away from the screen, away from your face, and then making a U-turn upwards this time, and then coming again towards the screen and towards your face to meet the superior ciliary body. So in the intact eye, these muscles form a continuous circle. We have just represented it as a cut section. So when that muscle as a whole contracts, you can imagine that the radius of the eyeball reduces. And when that happens, you can imagine the lens ligaments relaxing and the lens bulging anteroposteriorly. Now this is what we call accommodation of the lens. M3 receptors are present in the circular fibers of the ciliary muscle, and upon stimulation, the circular fibers of the ciliary muscle will contract. The lens ligaments will relax, and this causes the lens to bulge anteroposteriorly. Okay. Now, let us take a look at the sphincter pupillae now. So there's the sphincter pupillae of the inferior iris. Let's look at them together now. Again, doing what we did previously, let us try to do that now with the sphincter pupillae. Take a look at the sphincter pupillae of the superior iris. If this was not a cut section of the eye, and if the eye was intact, then you can imagine the superior sphincter pupillae emerge from the slide or emerge from the screen towards your face, and then make a U-turn downwards, and then move away from your face and towards the screen, and it will meet the screen at the inferior iris, and then it would pierce the inferior iris and move away from the screen and away from your face, and then make a U-turn upwards this time, and then move towards your face and meet the screen at the superior iris. Thus, in the intact eye, the sphincter pupillae is an intact circle, and when it contracts, the size of the pupil comes down. Finally, if you look at the longitudinal fibers of the ciliary muscle, these fibers are inserted into the scleral spur. When these muscles contract, when the longitudinal fibers of the ciliary muscle contract, they pull the scleral spur. and cause the iridocorneal angle to open up. And when the iridocorneal angle opens up, there is increased drainage of aqueous humor out of the eye, resulting in a fall in the intraocular pressure. So, as far as the eye is concerned, M3 receptors are present in the longitudinal fibers of the ciliary muscle, and upon stimulation it opens up the iridocorneal angle and increases the drainage of aqueous humor, resulting in a fall in the intraocular pressure. M3 receptors are also present in the circular fibers of the ciliary muscle. It reduces the overall radius of the eyeball, the lens ligaments relax, the lens bulges, and there is an accommodation of the lens. Finally, M3 receptors are present in the sphincter pupillae, and upon stimulation, it causes miosis.